So once we successfully purify that protein of interest, what do we do next? Well, logically speaking, if we don't know anything about that protein that we just isolated, what we want to do first is we want to actually determine what the amino acid composition is within that protein. So we want to know what types of amino acids are found within that protein and how many of each amino acid is found within that protein. So how do we calculate this information? How do we find this information? So this is what we're going to look at in this lecture. So we're going to begin by assuming we know what our protein is. So let's suppose we have a 6 amino acid polypeptide. So we have alanine, arginine, phenylalanine, glycine, aspartate, and glycine. Now, of course, usually you don't know what that protein actually is. But in this lecture, I give you the composition of that protein to basically show you how this method actually works. So once we have that beaker, and inside that beaker we have the solution that consists of only this type of protein, what do we do first? Well, ultimately what we want to do is we want to separate the amino acids, the different amino acids, into different compartments, and then we want to count up how many amino acids are found within each compartment. But to actually separate our amino acids, we first have to break the bonds holding these amino acids together in that polypeptide chain. So in the first step, we basically want to break the bonds. And the way we break the peptide bonds is we take this peptide, we place it into a solution that consists of 6 molar hydrochloric acid at a temperature of 110 degrees Celsius, and we wait for about 24 hours. And what will happen is that will hydrolyze and break down the peptide bonds, separating these amino acids. And so, following the addition of our peptide into this solution at this temperature for 24 hours, we're going to have a solution that consists of these individual constituent amino acids floating around in that water. Now, what exactly is the next step? So once we separated these amino acids, now we want to actually separate them into these individual compartments. So how exactly do we separate them into individual beakers? So we're going to basically use a procedure that we spoke about earlier. We're going to use the ion exchange chromatography technique. And this technique basically separates our molecules based on their net charge. So remember, these different amino acids will contain different side chain groups, and that means they will contain different properties such as the net charge. So we'll see exactly how that is useful in just a moment. So let's suppose that this is our setup. So this is the funnel placed on top of our column. Inside the column we have these specially formed gel beads that essentially contain a charge. So in this case we're going to use negatively charged beads. So this is our ion exchange chromatography technique, and we take the beaker that consists of these amino acids in their individual form and we essentially pour it into our funnel so that eventually they end up in this column. And because these have charges, they will interact with the charges found inside our column. And so what will happen is they're going to bind to different sections of our column. So for example, we see that alanine is somewhere here, we see that aspartate is here, we see that glycine is here, and so forth. Now, once these are actually bound, right, what will happen next? Well, the binding process differs for each one of these amino acids, and that's because they have different charge values and they have different types of side chain groups. And what that means is when we actually wash our column down with some type of material, when we wash them down, they will basically detach depending on what the concentration, what the pH, and what the volume is of that particular buffer. So we can use a buffer, for example, sodium citrate of increasing pH, to basically wash down our column. And as we wash down the column, these amino acids will begin to elute at different rates. So the identity of the amino acid can be determined based on the volume of the buffer and the pH of the buffer that is needed to actually elute that amino acid. So we essentially have these values that we can find in a textbook, and then we can actually determine what the values that we use are, and then we can compare them to those textbook values and see exactly what amino acid we are dealing with. So to see what we mean, let's suppose we use a buffer at a relatively low pH, and we use a very small amount of that volume before we see that one of those amino acids actually elutes. And so if we calculate how much volume we used and what the pH of that is, and we compare it to our textbook value, we'll see that the first amino acid was this green amino acid, which is aspartate, so D. Now, we continue our process. We continue pouring, and we increase the amount of volume, and we might change our pH, and then once we see that a second amino acid elutes, then we write down how much volume we used and what the pH of that buffer was. We compare it to our textbook value, and we see that it is this quantity here. So the x-axis basically describes the volume of the buffer that is added in order to elute that amino acid. So in this area, we have a very acidic buffer that we use. In this area, it's not as acidic. So as we go this way, the acidity of our buffer basically decreases. It becomes more basic. At the same time, the volume increases going in that direction. So we see that the second amino acid basically is our G, and that's glycine. Then we see that the next one in line is alanine, because this much volume is basically used at a specific type of pH. And we continue our process until we see that the next one is phenylalanine, and finally all the way here we have the R, which is arginine. And that makes sense because arginine is the most basic and aspartate is the most acidic. So step 2 basically allows us to actually separate our amino acids into different containers, and now we know what amino acid is found in which one of our containers. So we know which amino acids we have. The next question is, how many of each amino acid do we have in that beaker? So we want to count up those amino acids, and that's exactly what we do in step 3. So now that we know the types of amino acids involved, we must determine what the quantity of each one is in our polypeptide. And the way that we're going to do that is use a special type of molecule known as ninhydrin. So here we have our 5 beakers that we basically separated in step 2. So in beaker 1, we basically have arginine. In beaker 2, we have glycine. In beaker 3, we have aspartate. In beaker 4, we have phenylalanine. And in beaker 5, we have alanine. So we know which amino acids we have because we were able to compare to our textbook standard value as shown in this diagram. The next question is, what is the concentration? How many of these amino acids are found in each one of these solutions? So what we do is we take each one of these solutions, we basically add this special molecule known as ninhydrin. And what ninhydrin does is it reacts with that amino acid to form a special type of molecule that is able to absorb a special, a particular frequency or a particular wavelength of light. And for 19 of the 20 amino acids, it basically produces a very, very dark blue color or a very, very dark purple color. So we place the ninhydrin, it reacts with our amino acid, and then we basically heat it, and we produce a mixture. Now, once we heat that, we can then take a sample out of that beaker and we can place it into a special apparatus that essentially collects or determines how much light that solution can absorb. And we do that because After heating the solution of ninhydrin and the amino acid, we can test the solution for light absorption, because the higher the concentration of that amino acid is inside that solution that contains ninhydrin, the more light our solution actually absorbs. So for example, let's suppose we take this first beaker, we add ninhydrin to it, and we heat it. And so this will turn dark blue or dark purple. And then we take a pipette, we take a small sample out of that mixture, and we place it into a special apparatus that is able to absorb light, or determine how much light is actually absorbed. And so we shine light at a specific wavelength, it absorbs some, and we determine how much is absorbed. And we do it with every one of these beakers, and then we plot it on an XY axis, where the Y-axis is the absorption of light, and the X-axis is the type of amino acid we actually used. And so what we see is, the higher our value is, the higher this slope is, the higher our absorption is. And because in this protein we had one alanine, one arginine, one phenylalanine, and one aspartate, the height of these values will be exactly the same. But because we had two glycine amino acids, what that means is this will be twice as high as any other one of these slopes. And so what that means is we have twice as many of the glycine. So in this case, we have 1, 2 as these other ones. So we have 1, 1, 1, 1, and 2. And so in this manner, by using this step 3, we can basically determine what the relative amount is of each one of these amino acids in our protein. So now we know what the composition is, we know what types of amino acids we have, and we also know how many of each one do we have. What exactly is the next step? Well, in the next lecture, as we'll see, the next step is to actually determine what the specific sequence is of those amino acids inside that polypeptide chain.